Transcription is a fundamental process in molecular biology, where genetic information from DNA is converted into RNA. This step is crucial for gene expression and ultimately leads to protein synthesis. In eukaryotes, the enzyme RNA polymerase II plays a central role in this process. While the basic mechanism is highly conserved, recent research has uncovered fascinating variations, especially in yeast. Today, we'll explore how a distinct mechanism of transcription initiation evolved in budding yeasts and what this means for our understanding of gene regulation and evolution. Additionally, most eukaryotes use what's called the classic model of transcription initiation. Here, the transcription machinery assembles at a specific DNA sequence known as the TATA box, and RNA polymerase II starts transcription about 30 base pairs downstream. However, in the budding yeast Saccharomyces cerevisiae, scientists have discovered a different approach called the scanning model. In this model, the transcription machinery scans a broader region downstream of the TATA box, searching for the optimal site to begin transcription. This difference has profound implications for how genes are regulated in yeast. Understand these mechanisms, researchers map transcription start sites (TSSs) at single nucleotide resolution across 12 yeast species. Using advanced genomic techniques, they improved genome annotations and provided accurate boundaries for protein-coding genes. This high-resolution mapping revealed how different yeasts select their TSSs and highlighted the evolutionary divergence between species. Such detailed maps are essential for uncovering the genetic innovations that underlie new transcriptional mechanisms. By comparing TSS maps and genome sequences, scientists inferred when the scanning model emerged. The evidence suggests that this mechanism originated after the split between Yersinia lipolytica and other budding yeasts, more than 200 million years ago. This evolutionary event marked a significant shift in how yeast species initiate transcription, setting the stage for further genetic innovation. Key feature distinguishing the classic and scanning models is the distance between the TATA box and the TSS. In classic model species, the TATA box is tightly positioned about 30 base pairs upstream of the TSS, resulting in a sharp, focused promoter shape. In scanning model species, the TATA box is found further upstream, and the region between the TATA box and TSS is broader, leading to a more dispersed promoter shape. This difference in promoter architecture reflects the underlying mechanism of TSS selection in each group. Both classic and scanning models show a strong preference for pyrimidine, purine pi-pu dinucleotide at the TSS. This means that the first transcribed nucleotide is usually a purine, with a pyrimidine just upstream. This sequence preference is important for accurate recruitment of the first nucleotide by RNA polymerase II, which in turn increases the chances of proper mRNA capping, a critical step for efficient translation and stability of the transcript. Preference for purines at the +1 position of the TSS isn't just a random quirk. Research shows that having a purine here reduces the likelihood of transcription errors and increases the probability that the new RNA will be properly capped. This cap is essential for the RNA's stability and its ability to be translated into protein. Transcripts starting with a purine are much more likely to receive this protective cap, highlighting a direct link between sequence preference and functional efficiency. Striking discovery in scanning model yeast is the presence of an adenine-rich, or rich region immediately upstream of the TSS. This region, especially prominent in species that underwent whole genome duplication, appears to facilitate the selection of favorable TSSs. The air-rich region acts as a signal or anchor for the transcription machinery, helping it pause and initiate transcription at the right spot. This feature is largely absent in classic model species, underscoring its evolutionary novelty. Presence of an rich region upstream of the TSS is not just a passive feature. It actively influences which TSSs are used, leading to more efficient and accurate transcription initiation. In scanning model yeasts, most transcripts are initiated from TSSs associated with an air-rich region, while classic model species lack this enrichment. This suggests that the rich region is a key evolutionary adaptation that enhances gene regulation in these yeasts. By comparing closely related yeast species, researchers found that changes at specific nucleotide positions, especially at the -8, -1, and +1 sites, are closely linked to shifts in TSS usage and promoter activity. Mutations that disrupt the preferred pi-pu dinucleotide or the rich region can significantly reduce transcriptional activity or shift the TSS to a new location. This highlights the importance of these sequence elements in the evolutionary divergence of gene regulation. Beyond the TATA box and rich regions, yeast promoters contain other important sequence motifs. For example, the Ga element is enriched in TATLS promoters in scanning model species and may serve as a binding site for general transcription factors. De novo motif discovery has identified several other conserved elements that likely contribute to the diversity and regulation of transcription initiation in yeast. Researchers propose that in scanning model yeasts, the pre-initiation complex PIC assembles at the TATA box and then scans downstream, searching for an air-rich region followed by a pi-pu dinucleotide. When this combination is found, the PIC pauses and initiates transcription. This mechanism allows for flexible TSS selection and may reduce the production of unwanted transcript variants, enhancing gene regulation precision. Evolution of the scanning model in budding yeasts appears to have occurred in several steps. First, the mechanism for TSS selection shifted after the split from Y. lipolytica, likely due to changes in transcription factors and RNA polymerase II. Next, the rich region emerged as a new signal for TSS selection. Finally, in species that underwent whole genome duplication, a strong preference for adenine at the -8 position developed. Each step added new layers of regulation and complexity. Understanding the origin and evolution of transcription initiation mechanisms in yeasts provides valuable insights into the flexibility and adaptability of gene regulation. These findings not only improve genome annotations, but also shed light on the molecular basis of evolutionary change. Such knowledge can inform studies in other organisms and may have practical applications in biotechnology and synthetic biology. Summary: the discovery of the scanning model and its stepwise evolution in budding yeasts highlights the dynamic nature of transcription initiation. Key sequence elements like the pi-poo dinucleotide and a rich region play central roles in this process. Future research will focus on unraveling the detailed interactions between transcription factors and promoter elements, and exploring how these mechanisms operate in other species. The story of yeast transcription initiation is a testament to the power of evolutionary innovation.